From Microbe TV, this is Office Hours for Wednesday, October 16, 2024. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can learn all about viruses from a virologist. That would be me. Welcome, everybody, on this chilly night in New Jersey. Not sure what the temp is, but I bet it's in the teens Celsius. Let's thank our moderators for being here tonight. We have Barb Mac UK. We have Les. We have Andrew from New Zealand. And I thought I saw Frank, but maybe I didn't. It's a different Frank. Thanks, moderators, for being here tonight. Thanks, all of you, for joining us. There will be more soon. The plan for tonight is tell us where you're from first, so I can go through and say hi to everyone. Then I want to have a mini lecture on West Nile virus, which was inspired by Tony Fauci's opinion piece to the New York Times. And if you haven't seen that, I'm going to read parts of it tonight. Let's see who's here. Lisa is from Columbus, Ohio. They have 48C. Very chilly. It's the season, right? Summer's over. Rita, 55F in southwest Idaho. It's one of the few states in the U.S. that I have not been to. Idaho. Ramon, I love it. It's great. It should have been from Brooklyn. Will is from Changsha, China. 21C. Welcome. MN is in the DC area where it's 40s. Yeah, it's chilly, see? Risto is in Sydney, Australia, where it is too cloudy to see the comet. Yeah, we had a comet yesterday. Supposedly, I didn't see it. But yes, I was told it was a thing. Frank is from Santa Barbara, where it's 68F, which makes it dreary, I guess. Andrew's from New Zealand. Kang is from Chicago, 10C. No clouds obscuring the evening sky. Chris is in Montreal, Canada. Midge. I think Midge is in Australia, but I could be wrong. John is in the Twin Cities, where it's 55S with F, with fall colors everywhere. Krishnan is from Malaysia. That's pretty darn far away. Maureen is in Northeast Ohio. Looks like Tom has joined us as well from Western Oregon. Thanks, Tom, for joining us. Questella, it's 49 in South Indiana, Ohio. Vironauts. Yeah, I like vironauts. Micronauts, vironauts. It's all good. Catchy words are good. Alicia is in Belém in the Amazon region. 32C and dry. Margot is in East Phoenix, where it's 94. Only 94. Oh my gosh, it's so different. Sharon. I don't know where Sharon's from. Maybe we'll find out. Midge is in Sydney, Australia. There you go. Carol, our favorite nephrologist from Bakersfield, California, 82F. Elizabeth is in West Virginia. Is West Nile virus a flavivirus? It is. It's one word, flavivirus. Correct. I have scarlet and gray on, Ohio State colors. Yeah, I guess I do. The inside of this thing is gray. The outside is scarlet. I'm only wearing it because it's bloody chilly down here. I don't want to be chilly. I even have a space heater. I do have a fireplace behind me, but I didn't want to turn it on. It gets noisy and then it gets too hot and I have to turn it off. Blah, blah, blah. You know how it goes. Hat is in LA where it's 66F. Wherever Sharon is, it's 9C. And Alpine Sunny. Arizona, yeah, he never felt so sick. We're going to talk about that today. Asheville in Spartanburg, South Carolina. I guess you're okay. I'm sorry about the mess there. EG is for Mississauga, 7C in Brisk, Canada. And Tom is in the Oregon Coast Range tonight. Joseph is in Ontario, Canada. And here is Jonathan from Encinitas. Pat is from Nova Scotia. Oh, and then Sharon is from Tasmania. Okay. I didn't get it. You know, I'm thick. Well, I'm geographically thick. I don't know where a lot of things are. Aldo is from Peru. Welcome. Eden is from Montreal. Jessica is from Toledo, Ohio, where it's 47C. Mike and Lori Ding, Seattle, 15C. Hello, environmental, upstate New York, the southern tier. 40F. Welcome back. Thomas, hello. Where are you from? Margot is sitting on her patio in shorts. Oh my gosh. It's kind of that borderline weather where even around here you can see people in shorts, and there's some people who are just overheated. I knew a kid who went to high school the whole winter wearing shorts and a t-shirt. He never wore a jacket. I guess he had a high thermostat, right? Tom is in Salt Lake City, the home of Nels LD, where it is 75 degrees. And Peter is in Boston. Welcome, everyone. There's some good questions I've passed, and I will get back to them because I really want to answer them. But first, I wanted to talk about West Nile virus. And if new people join us, we'll acknowledge you later. So let's turn on the screen share. So this is called West Nile virus and Tony Fauci, inspired by Tony Fauci. I do teach a little bit about West Nile virus in my course, so I've used some of those slides here. So remember, West Nile virus is, or I should say West Nile virus disease. There is a name for the disease. For all virus infections, there's a name for the disease. Some of them are more interesting than others. So for West Nile virus, the disease it causes is called West Nile disease because it's very diverse. A lot of different things, as we'll see. And as you remember, we classify viruses as acute or latent. Basically, acute infections like rhinovirus, rotavirus, influenza virus, and West Nile virus. They are characterized by a period of virus shedding, which is shown here in the blue line, virus production. On the x-axis, it's time, and disease, which is the red bar. So it's a defined period Whereas latent infections, and there are a couple of different kinds or flavors, we've talked about these a bit. They go on for the lifetime of the host, like herpes viruses. You get them, and they're with you forever. No, you're not getting rid of them. Dr. So-and-so didn't cure your herpes. Sorry. So West Nile is an acute virus infection. And so some characteristics of acute infections: rapid onset of virus reproduction, limited but not necessarily short, but maybe severe course of disease, production of large numbers of virus particles, and eventually immune clearance. And the host either dies or survives and has some immunity. So that's an acute infection, and West Nile falls into that class. Here's the course of a typical acute infection, just to remind you what it looks like. So we have on the y-axis virus reproduction, and on the x-axis we have the duration of infection, or the time. So here at time 0 we have the initiation of infection, and the red line is the production of virus, right? And so it steadily increases after the initiation, and at a certain point you activate the innate immune system. It's very quick, most likely. In most people it's probably minutes to hours, and then the innate immune system kicks in, and most times the innate immune system actually clears the infection and you don't even know you had one. But other times it doesn't, for various reasons. Maybe your innate immune system isn't so good. Maybe the virus is has antagonizing proteins. The reproduction continues, and so then the adaptive response comes in, like antibodies and T cells. It takes some time for them to get going. They got to put their coffee away, get going, and check out the antigen. So in 7 to 10 days, they're kicking in. And many times in acute infection, the virus titers begin to decline just as the adaptive response is going up, which tells you that it's not making a big deal. I'm not making a big difference in the outcome of the infection. But of course, that'll provide memory that will help you next time, hopefully. Right? So that's a typical acute infection. And characteristic of acute infections are inapparent infections. All right? These are successful infections. The virus gets in you. It hits the cells that it will reproduce, and it begins to reproduce. But you have no idea because you have no symptoms. You don't feel anything. And there's no disease. So remember, a symptom is what only you can feel, like a stomach ache or fever. Though someone could measure fever, but initially only you know. I feel feverish, muscle aches, etc. Those are symptoms. You don't feel any in an inapparent infection. Now, the other part of this are the signs of infection, right? We talk about symptoms and signs. The signs are what anybody could measure. They could take your temperature, they could take your blood and measure viral particles or antibodies and so forth. And so an inapparent infection is asymptomatic. There are no symptoms, but there's certainly signs because the virus is in you and replicating. There's an immune response which you could measure and so forth. The problem with an inapparent infection is that you make enough virus particles to spread the infection. And so you're walking around, you feel just dandy, and you're spreading the infection. Some viruses are very good at that, others not so much. Like Ebola virus, you don't start spreading until you're sick. You really feel sick, you feel symptoms, right? But many other viruses, including SARS-CoV-2 famously, you start to spread the virus before you feel any symptoms. Now, how do we know this? How do we know there's so many inapparent infections? Well, we do serological surveys and we say, oh, look, 40% of the population has antibodies to this virus, but only 10% of them were sick. So the other 30%, there must have been some inapparent infections there. These are patterns of what I like to call well-adapted pathogens. There are viruses that have been evolving with humans for many, many thousands of years and have come to an equilibrium where the virus reproduces and can transmit to another host without making the host overtly ill. Not that I don't think that helps transmission. I think, well, of course it helps because you're spreading asymptomatically, but you would still transmit if only during the symptomatic period, but probably much less efficiently. And an example is poliovirus, right? Over 90% of poliovirus infections are inapparent. Everyone thinks of the paralysis caused by poliovirus. It's only 1% of infections. 90% have no symptoms, and only the 9% in between, non-specific symptoms that you couldn't attribute to a particular virus. And so during polio outbreaks in the US, in the beginning, they would quarantine kids who were paralyzed, which was useless because the people spreading the virus were not paralyzed because there are so many of them. And with SARS-CoV-2, we think maybe 40% of infections are asymptomatic. So it really helps for transmission. Okay, so those are acute infections. And these are a big problem, right? These are your big public health problems. Not that latent infections are not, but acute are because they cause big epidemics affecting millions of people each year. Norovirus, influenza virus, now SARS-CoV-2, respiratory syncytial virus, and many, many others. And the real issue here is that with an acute infection, by the time you feel sick, the infection may be over, the virus reproduction may be over, it's on the decline, and it's already spread. So you can't use sickness as a metric. You have to do something else. But we don't. For most cases, we feel a little crappy, and we say, go test yourself, and then you find out what you have, where it's probably already a few days late, which impacts antiviral efficacy, although it still works well. Okay? So that is, those are the characteristics of acute infections. Now, let's talk about West Nile virus. It's a flavivirus. And you should be able to rip off some other flaviviruses, right? Dengue virus, yellow fever virus, others. This virus was isolated in 1937 in the West Nile district of Uganda. It has nothing to do with Egypt. Yet, here this cartoonist looking at this patient, the doctor is looking at the patient who looks like an Egyptian with the headdress, and he says, we're pretty sure it's the West Nile virus. No, doc, you're wrong. It has nothing to do with Egypt. But people see Nile and they assume it's the Nile River. No, it's the West Nile district of Uganda. So this has been around for a while. We've known about West Nile since 1937. But it was out in the Western Hemisphere. Right, so here's this map showing the distribution currently of West Nile virus. Australia, Central Asia, Europe, parts of Africa, and now all of North and South America. And then there are other mosquito-borne viruses, St. Louis encephalitis, Japanese encephalitis, and so forth. So it was not in the Northern Hemisphere, the Western Hemisphere, till 1999, when suddenly in the summer of 1999, it was August, people started getting sick. They started getting fevers and muscle aches, and many of them got neurological symptoms. They got encephalitis and partial paralysis. And eventually, the virus was isolated and determined to be West Nile virus. It's a very interesting story. In fact, Dixon has a book that he wrote about it. I don't have a copy here, but it's called West Nile Virus. It's a small book. You can read it in one evening. It's very nice, recounting of the discovery. In New York City, besides these people getting ill, birds were dying at the Bronx Zoo. The exotic birds were dying, and that freaked them out. And a pathologist there, she looked at some of the dead birds and said, this is a neurological disease. There's something going on here. And so they eventually got the genome sequence. It turned out to be West Nile virus. Okay. And then, so it arrives in New York in 1999, and it starts to spread all across the U.S. It goes through all of North America and much of South America as well. Okay. And the mosquitoes are diverse, different kinds of mosquitoes, including culicine mosquitoes or culex species. Now, the reservoir are birds and rodents, and so they weren't infected in the U.S., for example, before 1999. But the introduction of the virus into the U.S. led to the establishment of the virus in a reservoir population, including birds and rodents. And many birds died because they'd never seen the virus before. It wiped them out. And so crows, blue jays were absent until they rebounded years later. It was really a murder of crows. Now, how did it get here? It turns out the sequence of the virus in New York is identical to a West Nile virus isolate from an Israeli goose farm. And so, how did it get here? Not clear. I doubt that a mosquito came over and survived long enough and bit someone, a viremic mosquito. People don't spread this to each other. So people don't have enough viremia, virus in the blood, to spread it to another person by a mosquito. So it's always mosquito transmitted. So a mosquito takes a blood meal of, say, a bird who's infected, and then later takes a blood meal from a human, and the human becomes an accidental host, and that's a dead end. And so every West Nile infection that you see is a spillover from a mosquito from a rodent or bird reservoir. And so just some coincidental occurrence in the summer of 99 brought the virus here. One of Dixon's theories is that a child came over on the plane who was infected in Israel. And he said maybe because the child's immune system wasn't so mature, the viremia was a little higher. And then when that child got to New York, it was bitten by a mosquito, and that started the cycle. But we'll never know. It's here, and now we have many, many, many cases since then. It spread across the U.S. in less than 4 years. Here's a nice map of that spread. So the yellow shows you the states that were positive. So initially, 99, New York, and it begins to spread around New York and then down south and then more and more and more. As you can see, by 2008, most states, and now every state, I believe, has had West Nile virus. By October 2004, 1 million people were infected. We know that from serological surveys. And febrile illness developed in about 20% of people. But in 1% of infected people, they got central nervous system disease like paralysis and cognitive and motor defects and so forth. So it's not an insignificant disease. And many people were infected with no obvious disease. And so, I say here, it's hard to stop an epidemic, but it really wouldn't matter because it's not the people who are spreading the virus. It's the reservoir, the birds being bitten by mosquitoes. This is true for most other acute virus infections, but not here. Okay, so this is, again, a flavivirus, an envelope plus strand RNA virus spread by many different kinds of mosquitoes, including Culex species. And here is the transmission cycle. So its reservoir is primarily in birds, and mosquitoes transmit the virus from bird to bird. Yeah, birds get bitten by mosquitoes. So do rodents and other animals in the forest. And so if a bird is viremic, the mosquito picks up virus. When it bites another bird, it transmits it. And so that's the cycle that maintains virus in the environment. And these birds are generally okay. It's only when you have a bird never seeing the virus before it gets very sick like they did in the U.S. Now, occasionally one of these mosquitoes will bite a human or a horse, in fact, and they get infected. They can develop West Nile disease. And they're dead-end hosts, right? They do not transmit the virus to anyone else. So a West Nile infected person is no transmission threat. Nevertheless, you have to work on the virus at BSL-3 conditions, which makes zero sense at all. Because this is not something you can accidentally inhale, or you could certainly do a needle stick, but that shouldn't warrant a BSL-3. Right, so humans and horses get very sick and they can die, and that's unhappy for the horse owners because they're expensive, right? So we actually have a horse West Nile virus vaccine, but we don't have one for humans. Huh. So culisine mosquitoes bite and transmit the virus. The incubation period is 3 to 14 days. 20 to 30% of infected people develop a flu-like illness called West Nile fever, right? Malaise, muscle aches, so forth. And 80% of people have no symptoms whatsoever. Yet they're infected, and you can show by serological tests that they've made an immune response to the virus. The real problem here is that about 1 in 150 people develop what we call neuroinvasive disease. The virus gets into the central nervous system, the brain and spinal cord, and you get headache, you have eye issues, muscle weakness, cognitive impairment, a polio-like flaccid paralysis. And this is accompanied by a 10% mortality, not insignificant. And over half of these individuals with neurological symptoms have long-term neurological sequelae. It's kind of a long West Nile, right? They never get better. You'll come back to this, you'll see. Here's a kind of a scheme of the pathogenesis. So at the top, we have the virus being picked up by mosquitoes from birds, and the mosquitoes biting a human. The virus is picked up by dendritic cells, which is part of the sensing mechanism, and they're brought to the lymph nodes, and they reproduce in lymphoid cells. They also can the virus can then make its way to the spleen, where it finds more lymphoid cells and reproduces even more. So lots of virus in the blood. And then, interestingly, the virus is sensed by the innate immune system, in particular by toll-like receptor 3. And one of the products of innate immune sensing is the production of tumor necrosis factor alpha, which is meant to kill virus-infected cells. But what it also does is permeabilizes the capillaries in the brain so the virus can get into the brain. It's an unintended consequence, I assume. And in the brain, it can affect neurons, giving you those neurological symptoms. All these other cells here in this picture are those that we believe are important for control, including B and T cells of different sorts, complement, interferon, and also cells in the brain. Okay, so here's a graph of West Nile in the U.S. Up until 2019. So we have from 99 onwards, we have the number of disease cases, neuroinvasive, non-neuroinvasive, and total cases. So let's look at the total cases here. 62 that first year, and in some years to 9,000. 4, 3, 5,000, up and down, right? Some years 700. And now that it's established, we don't seem to get below 700. And so, folks, as you're listening to this is here in New York, New Jersey, wherever you are. It's there. So be careful. Protect yourself from mosquitoes. Look at the number of deaths. I mean, this isn't huge, but it's substantial, right? We'd like to prevent that. And then neuroinvasive versus non-neuroinvasive. A lot of neuroinvasive cases, as you can see. That changes from year to year. All right, so we'll come back to this. This is an important graph. So when I look at this, I say we should have a vaccine. Okay? Keep that in mind. So here's some statistics. This is neuroinvasive West Nile virus disease by state, 99 to 2019. And the dark ones have a lot. Incidents per 100,000, the dark are greater than 1. Why it should be higher in some states, I don't think we know, but as you'll see, age is a risk factor for neuroinvasive disease. So that could be part of it in some of these states, but it's not all, obviously. So this is the disease reported by week of illness onset. So we have the number of cases on the y-axis. In the week of the year, you can see they start to go up in June, they peak August, September, and pretty much gone in December. They follow the mosquito cycle, obviously. So these are the months you have to be careful. You really need to be careful because you never know when you're going to get it. And then here's the big one, neuroinvasive disease by age group. When Tony Fauci got sick, I put this up on Instagram right away because he's 80 something, right? He's in the highest risk group. So here is the incidence of neuroinvasive disease per 100,000 with different ages. And you can see it steadily ticks upward with age until over 70, you have a really high risk, almost 1.4 cases per 100,000. Much higher than at a younger age. All right. So how do we prevent it? Well, we know these species bite from the evening to the morning. So if you're going to be out and about during those hours, you should have repellents. You should wear clothing everywhere. Tuck your pants into your socks, especially if you're going out in the woods. Definitely have screens on your homes. We don't have a vaccine. We do have a vaccine in horses because, why? Because they're economically important. But we don't have one for people because I guess we're not economically important. So West Nile infection, 30% fatality in horses. It's much higher than in people. But no West Nile vaccine for people. And that's weird because we are able to make a vaccine. Since 99, we should have made a vaccine. I definitely think that's a shortcoming. All right, so that brings us to Tony Fauci. So just beginning of October, he publishes a guest essay in the New York Times. Mosquito in my backyard made me the sickest I've ever been. And you know that Fauci was the former NIAID director. And let me just preface this by saying he has a big say in what vaccines are developed. Okay? Now, let's read a little. After I spent more than 50 years chasing and fighting viruses, one fought back and nearly took me down. I speak of the West Nile virus, delivered by the deadliest animal on the planet, the mosquito. I didn't get infected during any of my international trips over the years, but most likely I was outside my home in Washington, D.C. In mid-August, I was feeling weak and exhausted, but I attributed it to a recent bout with COVID-19. Though I had tested positive for COVID over a month prior, I had experienced a rebound of symptoms. I very much object to the use of rebound. It's a natural part of the disease progression. After taking the treatment Paxlovid, perhaps I was still experiencing lingering symptoms that would eventually resolve. But not so. Instead, I began to experience unexplained severe fatigue and exhaustion, culminating in my admission to a hospital on August 16th, delirious and incoherent with a temperature of 103 degrees. I remember little of the 5.5 days that I spent in the hospital, except that I had never felt so ill in my life. My physicians assumed that I had sepsis and treated me with antibiotics. After several days, my fever subsided. I was discharged on antibiotics without a clear diagnosis. That changed the next day when blood tests revealed that I had West Nile virus. Kind of slow on those blood tests, right? Should have been the same day, but they weren't looking for it. There was no treatment for West Nile, and I was left to deal with its toll on my body. It was terrifying. I could not swing my legs over the side of the bed to sit up without help. I could not stand up without assistance, certainly could not walk. A scary part of the ordeal was the effect on my cognition. I was disoriented, unable to remember certain words, asking questions of my family I should have known the answers to. I was afraid I would never recover and return to normal. Fortunately, over a period of a few weeks, slow improvement began. I was able to walk. Now I can walk a few miles with only minimal fatigue. My cognitive issues have completely resolved. I am on my way to a total recovery, but it's been a harrowing experience. I tell my story because West Nile virus is a disease. No, actually, Tony, West Nile virus is a virus. West Nile virus disease is a disease that for many, but probably the editors made them take out the two diseases, that for many people can have devastating and permanent consequences. I would have written this. I tell my story because disease caused by West Nile virus can have for many people devastating and permanent consequences. All right? That's the way to write it. At my age of 83, I was at risk of permanent neurological impairment and even death, yet the public may be unaware of the danger of this disease and that it continues, that little typo there, continues to spread across the U.S. It's been identified in 46 states this year. Unfortunately, very little is being done about it from scientific and public awareness perspectives. So Tony feels the brunt of a virus being ignored. So he says it's a flavivirus, came to New York in 99. Since 99, 60,000 cases have been reported, surely higher, maybe millions. 30,000 have had neurological symptoms, 23,000 hospitalizations, and close to 3,000 deaths. Now, here's the thing. He said, I understand the challenges for research. Having been director of NIAID, when West Nile was first identified, we began efforts to develop a vaccine. However, we could not move the trials beyond early testing, in part because the number of cases varied from year to year. For example, 700 2011 and more than 5,600 a year later. Such fluctuations make it almost impossible to have a consistent enough number of cases to show that a vaccine was effective at preventing infection. Same applies for antivirals. So pharmaceutical companies lack the financial incentive to make major investments. Aha! But what about NIAID? They made major investments in COVID vaccines, right? You said, let's do that, but you didn't think West Nile was important enough. Now, he uses the excuse that there weren't enough cases, but I'm not buying it. So he says, well, how do we deal with this? We have to do international trials. Can't be just U.S. We have to have global partnerships with the NIH, and we should do this for West Nile. Well, Tony, why didn't you think about it in 2000 when you decided you couldn't make a vaccine test here because of the numbers? Because it took Tony to get sick to realize how bad this can freaking be. And that's unfortunate. The director of NIAID should not do that. He or she should say, this is a real bad disease and we need to deal with it. So I just think that I agree we should have a vaccine. It should have been done years ago. And I think Tony probably is responsible for not doing it. Okay, so he says there's too much fluctuation in the number of cases, okay? So let's look at that. Yes, 4,000, 9,000, 3,000, that's fine. Only a few years there's 700. You got a lot of years right in a row where there are thousands of cases. I'm not buying that excuse, okay? I am not buying it. Plus, let's look at polio. These are polio cases in the U.S. since 1910. There weren't huge numbers every year. It went up and down, right? Look, 16,000, a few thousand, 10,000, 15, a few thousand, up and down, up and down. And they made a polio vaccine. Well, Tony Fauci wasn't involved in the decision. They decided this was a problem. Paralysis is a bad thing. Same thing with West Nile-like paralysis and neurological issues. It's a bad thing. And that's why you want to make a vaccine to prevent severe disease. Not to prevent infection, to prevent severe disease. Okay, so that's my feeling on this is Tony got awakened by his own vulnerability. That's fine. But he really should have made the go-ahead for a West Nile vaccine years ago because it is severe. You got enough cases in the U.S. to test the vaccine. I'm sorry. And if you wanted to, you could go overseas. And it was his decision not to do that. So I put the blame largely on him, but I'm sure others were involved as well. So that's what I wanted to tell you this week. West Nile virus, a little biology, and how Tony was involved with it. All right, let's go to some questions here. How prevalent is West Nile in Africa? What a good question. What a good question. Well, it's been circulating there for years. It's endemic. That doesn't tell us how prevalent it is, right? You want numbers. It's been detected in 20 African countries, also domestic animals. I'm not finding, in Madagascar, 30% of the oxen are positive. I don't know what the numbers are. But I think that you can't complain. We didn't make a vaccine when you were involved in not making it. And now all of a sudden you have a revelation because you got sick. I just don't think that's right. Environmental says perhaps Tony couldn't get the funding, or did he just not try because it was not statistically significant to him? I don't know what he was thinking, right? He probably thought this is not such a big deal. But I'm guessing. No, he could have gotten the funding. He was head of NIAID. He can get funding for anything he wants. Yeah. Come on, Tony. Right. Come on. Okay, Nushariza wants to know why a human is a dead end. Does this mean the mosquito can't do transmission from human to human for this virus? That's exactly right. So once the mosquito delivers the virus to a human, it reproduces in the human, and it never transmits to another human, neither by a mosquito nor by human to human transmission. That's exactly what it means. Yeah. What would not be funded if he worked on West Nile? It's his problem. He's the director. He's got to deal with tough questions, right? I mean, a lot of people die and have permanent disability from West Nile. We have the capability to make a vaccine. Actually, his last sentence in the op-ed, which I didn't show you, is something like, we have an obligation to public health to ameliorate disease, and he didn't do it. Oh, by the way, big farmers also at to blame because, understandably, they don't want to invest in something that they don't think is going to turn a big profit. The West Nile vaccine for horses turns a profit, so they're okay with that. But they looked at the numbers for West Nile and people, and they said, no, it's not worth it. Right now, Pfizer and Moderna are not doing so well with their COVID vaccines because there's already a little uptake, okay? There's a problem with vaccines, and it probably tells us that we should have a different way of developing vaccines than we do now. Polio vaccine was not developed by pharma. Eventually, it was manufactured by them. But Salk and Sabin developed their own vaccines. Here we got some numbers from Pete, Italy. 329 cases, 26 deaths, circulating in 10 regions. You're fired. Only Tony's already retired. That's right. That's very good. Very, very good on that. I'm just looking for the West Nile questions, and then we'll get into the others. John says maybe if we invested in a West Nile vaccine, it would also have unexpected benefits just from doing the research. Totally. I totally agree with that. Pete said, yeah, polio, mainly children. West Nile, the elderly, West Nile neuroinvasive disease, right? So you're talking about polio and neuroinvasive disease. That's right. It's interesting, right? Clothing is probably the easiest sort of management. Yeah. But people don't follow the advice, right? Because obviously they keep getting bitten. Whereas Costello said they can bite through clothing sometimes. Yeah. Can happen. Tryon wants to know, did cryoglobulin levels increase after West Nile infection? I really don't know. I have no idea. Makes me wonder just how comprehensive our surveillance systems are. So for West Nile, it's a reportable disease, right? But there are mosquito surveillance programs in multiple states. Um, and you can go find those data online. You can see the percent of mosquitoes testing positive for West Nile with time. It's actually not bad, but it's not great, right? Yeah, so Questella says you also don't tend to put stuff on when you run out for a minute. And yeah, then you get bitten. I have to avoid being outside much of the summer because it's so bad. Going to try the mosquito drops next year, see if it cuts down at least. And of course, if it's hot, 90-degree heat, you pass out. That's another issue. Does interferon protect against neuroinvasive? I would guess that if you got it early, but I'm not aware of any trials for that. No. Now, this Questella gets bitten on her face or eyelids. She votes for the vaccine. Hmm. Are they planning to release modified mosquitoes? I don't know about the U.S. I know other countries have already started to release modified mosquitoes for dengue, but for West Nile, let's look. There's always looking it up. It says it's being used in the U.S. to control mosquitoes that transmit Zika virus, dengue, chikungunya, and yellow fever. But not West Nile. It may be very hard because there's so many different species, right? But I don't know of any. Yeah, the first genetically modified mosquitoes were released in the U.S. in 2021. It's a company called Oxitec in Florida. They got a lot of pushback, as you might imagine. People didn't want genetically modified mosquitoes released. People don't want to be genetically modified. They don't want to eat genetically modified stuff, whether it's plant or animal. And they don't want you to genetically modify anything. Good luck with that. But I do think releasing modified mosquitoes that can impair transmission is great. And we're at early days in seeing the efficacy of that. We had a great 12 years ago with some people from Microsoft who designed this trap, a mosquito trap, which was flown by drones into remote areas, and the doors on this thing would open, and mosquitoes would fly in, and the traps would close, and they'd bring them back to do mosquito surveillance. It's very cool. How does West Nile reproduce? Oh, there is a receptor. I just didn't have it on that slide. I don't even know what it is actually. But yes, there is a receptor for sure. John wants to know, are Culex mosquitoes everywhere? Or did they spread here in 99? No, the mosquitoes were already here. You're imagining a bird? I guess it's possible, John. I have no idea. People who work in the airline industry may have a sense of whether that ever happens. Is the compartment pressurized down there so that the bird would survive? I guess they put animals down there sometimes, so it must be, right? That's pretty funny. What's the mortality? 10% of the neuroinvasive or the total? Well, let's see. So it's the total. Yeah. It's a total, not just neuroinvasive. Can you get long West Nile similar to long COVID? Well, good question. So I guess the neuroinvasive sequelae that don't clear up are a kind of long West Nile, right? So if you have lingering cognitive issues and lingering motor problems, that would be a long West Nile. Yeah, I was thinking about that also. Can you get West Nile twice? Let's see. Google is filling in my question. Yeah, some people do if they are immunocompromised or immunosuppressed in some way. But they say most people do not get infected again. So good idea for a vaccine, right? Okay. Yeah, Google says no West Nile currently in Britain. Yep. Hey, um, yeah, the climate change is going to change mosquito patterns everywhere. But these are present throughout the U.S. already. It's not like Aedes aegypti, which is not, and with warming will increase its range. This is already here. Betty is in Washington Heights. Welcome. Welcome. All right, let me go back to the top. There were some interesting questions up there. Okay, here's the first one. Will writes, yesterday, China's CDC released COVID stats for September for the whole of the country. It stated there were 6 deaths. The U.S., with a quarter of the population, had 5,000 deaths. I don't believe it. Hard to believe, right? But, I mean, the population isn't any younger than the U.S., is it? That's a big risk factor for death. But, this is not something you should lie about. Public health, if you start lying about public health, then you get in big trouble. So I'm hoping there's an explanation. And I doubt that's because they did mass vaccinations, blah, 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 and so forth. But thanks for letting us know. That's very interesting. Huh. Okay, here's another one from Frank. Is the relationship between SARS-CoV-2 and B cells different from that of other viruses? Is it different for the vaccine? This is a great question because we just did a relevant paper on immune yesterday. So I actually have it here. Let me share it with you. So this is a Nature Medicine paper. SARS-CoV-2 specific plasma cells are not durably established in the bone marrow long-lived compartment after mRNA vaccination. So the idea here is that when you get infected or vaccinated, your local lymph nodes encounter the antigen. They make cells called antibody-producing cells, and those antibody-producing cells make antibody for a while, and most of them eventually die, but some of them go into the bone marrow and are conferred with living forever, basically, long-lived, many years, decades. And then those are your memory B cells. And so when you get another infection, within a few days, they pop out of the bone marrow and they start making antibodies and take care of preventing severe disease. It looks like from this paper that either infection or vaccination by SARS-CoV-2 does not lead to the formation of these long-lived bone marrow antibody secreting cells. And their controls were very interesting. They only had 19 people in this study. Their controls, they looked at antibodies to tetanus toxin and influenza. And they found high numbers, good percentages of long-lived antibody-secreting cells in the bone marrow for those two, but not for SARS-CoV-2. And it didn't matter whether you're infected or vaccinated and infected, you didn't have any. And this was, they looked up to 3 years after the vaccines and still no long-lived cells. Now, the question is, so it's obviously not, it's not universal because flu and tetanus vaccines do induce these long-lived cells. And again, we're talking about B cells that make antibodies. So what's going on? One thing you might say is it the mRNA vaccines? Well, they looked also at people who got adenovirus vectored vaccines. So they had the same issue. So it's not the vaccine formulation. It could be the protein itself. Maybe the protein is structurally different in a way that doesn't induce long-lived B cells. And I think it would have been interesting in this study had they compared people who got Novavax protein-based vaccines, right? Now, remember, the common cold coronaviruses, SARS-1, MERS, none of those induce long-lived memory B cells. So this may be a general feature of coronaviruses, that their antigens are just not good at making long-lived memory B cells. So maybe we could improve that. Maybe we could change the vaccine in some way to improve that. But here's the thing. This is the outcome of it. We're chasing variants now here in the U.S. Anytime a new variant pops up and it starts to dominate, we switch the vaccine to cover it. I don't think that's relevant at all. I think the problem is that we don't make long-term memory B cells. Our antibodies go way down and we have no chance. So even if you match the variant within 3 to 6 months, that's what the kinetics were in this paper, you have zilch antibodies left in the serum. So you have no chance whether you're chasing a variant or not. We've been blaming the variants, but in fact, it's not. It's the vaccines, well, the infection, and they say the antigen most likely, but we don't know what it is yet. I hope that answers your question. So that's a good one. And I thought that the discussion was interesting. I think the immune will come out next week. Very cool. Okay, Chris in Montreal. COVID-19 disease and our mRNA vaccines and cardiovascular health risk. So from everything I've heard from Daniel, cardiovascular risk with mRNA vaccines is low, predominantly myocarditis, which is treatable and usually resolves. And it's much higher with natural infection. So the risk benefit is clear in favor of being vaccinated. However, you can get Novavax, but yeah, it's not available in some places. Hopefully that will change because that's not associated with it's actually still a low rate of myocarditis, even with Novavax. So it seems to be a property of the spike protein. I'd really like to know if Novavax makes long-term memory B cells in the bone marrow. But they didn't do that. That's a good question. Okay, Luminous asks, where do you stand on adults who don't have immune-compromised systems getting the new COVID vaccine? Well, as I said, I don't see the evidence that having a matched booster for the circulating variant is all that useful. We never get protection data. No matter what vaccine you're getting, what kind of COVID vaccine you're getting, the antibody level titers crash within a few months. And so you're going to get infected again. And even if the variant hasn't changed, you're not going to be protected. So, if you want another 3 to 6 months of protection, you could get it. On the other hand, if you're healthy, I would say if you're under 65 and healthy, you don't need it. And I think that's in line with CDC recommendations. And it's in line with what Daniel's recommending. So I don't want to give you medical advice, but scientifically, I don't see evidence that the booster really helps reduce severe disease as opposed to not having any. So here's the thing: when you get, let's say, 6 months out after a vaccine, your antibody levels have crashed, and now you get infected with SARS-CoV-2. You're going to get infected, and you're going to get some mild or maybe moderate disease. But if you're healthy, your T cells will kick in, and they will prevent severe disease. Now, I would say if you're over 65 and you test positive, you should consider taking Paxlovid, right, an antiviral, because that will resolve your severe disease rapidly and doesn't require you to get a vaccine. But I understand also that's an availability issue. It's also expensive. So there are lots of issues that play in here. I personally, I'm not getting the latest COVID vaccine. I don't think it helps, even though I'm over 70. Any comment on Dr. Sete's paper on previous flu vaccination, possibly blunting human disease with H5N1 via cross-reactive T cells? Yeah, I did see that. I think it's good, right? It's good that it would blunt potential H5N1 disease, but I don't think we should depend on that in case H5N1 starts circulating, which it has not yet, right? And we don't know if it will. So I think it's good, but it's not the solution, right? Hello, Vironauts. I should put a thing here. Let me put one up. Show in all scenes. Let me put up a new text things. Hello, welcome, Vironauts. We like Vironauts, right? Look, it came with a nice border and everything. We'll have that show up everywhere. Welcome, Vironauts. Naut, like spoken as a Jersey boy. Vironauts. All right, what else do we got here? Rita says a couple of local friends, remember this is Idaho, contracted West Nile and became seriously ill, encephalitis. Yes. You don't want to get West Nile, especially if you're older, right? So be careful. Eden wants to know, can you explain the difference between a plaque assay and focus forming assay for lytic and non-lytic viruses? Okay, sure. So a plaque assay, you take cells in a dish, you infect them with virus, and you cover the cells with an agar overlay. It's a semi-solid overlay. And after a few days, the viruses make holes in the monolayer because they're killing the cells, which you can take the monolayer off and you can stain and see nice holes, which you can count. And now you make it one plaque is a plaque forming unit. So you quantify infectivity as plaque forming units per mil, right? And many viruses are not lytic, or they don't kill cells so that you get a nice plaque. So there you can use a focus forming assay. So there are different ways you can do this. You could do the plaque assay as usual, and then at some point add antibody to the virus in the overlay, and then somehow stain the antibody, and the antibody staining will give you nice plaques. So even though the cells aren't dead, the antibody is searching for the presence of viral proteins, and you can add the antibody and a detector, and you'll get nice round circles of whatever color caused by that staining. So that's a focus forming assay. There are other ways to do it as well. Also for retroviruses that transform cells, they make them morphologically different. You can count different morphologically changed piles of cells on the monolayer. You can also put a gene in the virus that makes a color. And so even though you don't get a nice killing plaque, a cell killing plaque, you will get a colored region that's circular like a plaque because of the dye, and you can count those as well. So lots of tricks that you can use when your virus doesn't kill cells and it doesn't cause plaques. Yeah, good question. These are the kind of geeky questions we love. Because, well, first of all, they have clear, straightforward answers. Oh my gosh, some questions, they're not like this one. Next one from Kang. Oh my gosh. Does R naught decrease as more of the population becomes vaccinated or survives an infection? All right. I used to like R naught. So R naught is a number which tells you if a person is infected with a virus, how many other people on average can that person infect? Given sufficient time of contact between the two. And so high R naught is, so if you have an R naught of 15, on average, you could infect 15 other people. And if it's low, if it's 1, it's hard to sustain an epidemic. And if it's below 1, the epidemic will stop. So it's used by epidemiologists to try and get a handle on when epidemics will end. And so, yes, we think that well, here's the problem. Does vaccination stop transmission? I don't think anyone has ever shown it stops people getting disease, but it's not the same as transmission, right? Because what about polio? Yeah, vaccination gives you 0 cases of polio, say. But how many people are still infected? Nobody looks. You just assume that because it's preventing disease, it's also preventing infection, which has happened over and over again. People have this misconception. So I don't think we have any good evidence that vaccines stop transmission. Maybe they reduce it. So that would, in theory, reduce the R naught. But, there's so many factors that go into R naught that are independent of the virus. And so, the number of people getting together, the length of time they're together, what kind of situation, are they in a closed room, or are they outside? All that will affect the R naught. So maybe you could figure out an R naught at the beginning of an epidemic when there's no immunity. Yeah, because immunity can affect R naught as well. At the beginning of an epidemic when there's no immunity and the virus hasn't transmitted much. And so sometimes people do that. But the problem is then at different points in the epidemic, they'll recalculate it. So, we have a new variant. Let's calculate the R naught. No, you can't do that. No longer an R naught. But they do it anyway. They get people scared. Oh, this looks like it's transmitting better. Well, it may have nothing to do with the virus. So, long answer to your short question. In theory, the R naught should decrease with vaccination or immunity. But I don't think it's as clear as that the epidemiologists would like us to think. It's not a biological constant, let's put it that way. It's more or less a relationship. Theodore's from Athens, which is, the country. Look at the flag there. Very good. Thanks for showing that. And CYP3 is in Sweden. We're at 7C. Wow. CYP3, the target of ritonavir. Jessica says, so latent is the same as a persistent. So latent is a kind of persistent infection. So persistent just means that it lasts the lifetime of the host, right? A latent infection, and that can be shedding virus all the time, or it can be periods of virus and nothing, which would be a latent infection. Latent means the genome is still there, but you're not making virus, like a herpes simplex latency. So it's just a form of a persistent infection. We use words to name all different things. We just need to be consistent about them. Otherwise, people get confused. Environmental got Lyme this week. She got the ring rash. Yeah, so if you get a tick bite and you get a ring rash, take Dox. You'll be okay. Yep. Okay, Eden wants to know, do all pathogenic viruses, lice cells, have a lytic phase? So the answer to that is no, they don't all lyse cells. Some of them are non-lytic or non-cytopathic, as we say. And so you might say, how do they get out of cells? Well, there are various ways you can get out of a cell without killing it, right? If you're an envelope virus, you can just be released in vesicles and not kill the cell, and many viruses do that. And even non-envelope viruses can get released in vesicles without killing the cell. So, in your second question, knowing that latent viruses can reactivate to infect other cells, is that reactivation always going to damage the cell upon release? So, no, it won't always damage the cell. This is a good question for herpes. So, herpes simplex 1 is latent in neurons of the peripheral nervous system, right outside of your brain and spinal cord. And periodically, under stress, the virus begins to replicate and produce infectious viruses. And the question is, are they killing the neuron that they're replicating in? And I've asked many herpes virologists, and the consensus appears to be yes, but we don't know how many. And obviously, you know, you can live your whole life with herpes and get multiple reactivations, and you don't have any neurological issues. So probably not enough for damage to cause a problem. Yeah. Tryon wants to know, can immune tolerance induce persistent infections? Yeah, Tryon, you ask a lot of questions that I can't answer. All right. So, yes, I mean, in theory, the answer is yes, right? Because if you're infected with a virus that is recognized as self, that is not going to lead to clearance, and you're going to get a persistent infection. Yeah. But it's not black and white. It's not like total immune tolerance. It's got to be partial to one or a few antigens. And I can't tell you an example of that. All I can tell you is that it logically makes sense, right? Because most persistent infections start as an acute infection that's not cleared for whatever reason, immune antagonism, or the virus is in a immunoprivileged site. And so, yes, immune tolerance could be another mechanism for that, for sure, for sure. Martha is in Charleston. It's going to go back and listen. Yes, you should listen to it. It's good. Do persistent infections, is the other question, induce immune tolerance? Yeah, they could. Well, in my lectures, I'm trying to pull from my lectures answers to your questions. And I can't give you an answer about a persistent infection that induces immune tolerance. So I'm going to look it up. Yeah, so actually, hepatitis B virus may be a case where, so if you're infected as an infant, first 5 years of life, you're more likely to become persistently infected for life. Whereas if you're infected as an adult, you can clear the virus and get rid of it. So what's with the kids? And one of the theories is that the they get tolerant. The children's immune system get tolerized to hepatitis B virus proteins. And there's some specific mechanisms involved. In fact, we did a TWIV on this, which at the Chicago HBV meeting recently, which is going to be released in a couple of weeks, and they go into that in some detail. So yes, there's an example of how, in one case, hepatitis B infection induces immune tolerance. Folks, 147 of you. That's great. Please hit the like button. We've got 92 likes. We'd like to try and attract more people to learn a little bit about virology, maybe to make more informed health decision and not be scared by people who are passing on misinformation, to help you distinguish real stuff from misinformation. Midge wants to know if I asked Daniel about NAC. Yeah, I did. He says we need more data. It's not clear that it's effective. But I did a couple of weeks ago when you asked. Yeah. Eden wants to know if I could explain the lipid stalk mode. I don't know what that is. With respect to what? I've actually never heard of it. And also, what is the significance of the GDD motif in RNA-dependent RNA polymerase? That I know. GlyAsp-Asp, 3 amino acids, is a signature for an RNA-dependent RNA polymerase. It's 3 amino acids that are present in the active site of the enzyme that play a crucial role in catalysis. In other words, in making new chains of RNA. So every every RNA polymerase has a GDD motif. And that's how you can identify. If you have a protein, let's say you're sequencing the ocean and you find proteins with GDD, you can be sure that they're likely a RNA polymerase because of that, because it's essential for catalysis. That's the significance of the RDD. If you change any one of those amino acids, and this is an exam question every year in my class, what happens if you change one of those amino acids? Well, what happens is the enzyme's dead in the water, can't work, can't make RNA. So it's essential. Yep. Anyway, and apparently they've been releasing modified mosquitoes in the U.S. And they will continue to do so. But, they get a lot of opposition in in various states because people don't like the idea. They think these mosquitoes are going to get out of control. So it's all about informing people, right? There was a mosquito release in Florida, in Australia, near Cairns, I think, a couple of years ago. And they did a long-term public engagement program to tell people what they were going to do, why they were doing it, potential outcomes. And, they would have meetings and talk to people. It took time, cost money, but it was essential and they got it approved. And that's what you have to do. You can't just go in and release mosquitoes. People are not going to approve it. I was in Singapore in 2019, and they release mosquitoes regularly to control dengue. They brought me to the mosquito growing factory. It's so cool. They separate male and female mosquitoes, and they just release the males that are they're modified. And they release them and they're sterile, so they eventually wipe out the population. But they engage the community in doing this, right? They have the community go out there and they teach them what's going on. They have a whole program at the center that I visited where they bring people in to teach them. And then they have people involved in the release. They give people buckets of mosquitoes and they say, here, bring this to your floor. So many of the apartment buildings in Singapore are very tall. They have the elevators open on the exterior, and there's a whole walkway on the outside. So they have plants up there, and mosquitoes go all the way up there, 50 stories up. Mosquitoes. So you have to release these modified mosquitoes on every floor. So they involve the community. They go by their school and release mosquitoes. So, it's like any biological. You can have a great biological, but if you can't convince people to take it or use it, it's not much use. And it's extra work that maybe you didn't sign up for as a scientist, but now you're signed up and you have to do it. Angry Penguin. Are there any antibody rapid tests for home use that are not currently available, but you wish were available? I don't want people to take antibody tests because it's not clear what it means. I've had many people here, for example, saying, "Oh, I did a measles antibody. I have this level. Should I get a vaccine?" Don't know. We have no clinical data on what those numbers mean. So I think it gives you information that's not useful. So I don't want to have that. And remember, an antibody test will tell you if you've been infected in the past, right? So I don't want you. I think if you'd like to know who's infected now, in other words, an antigen test, a rapid antigen test, that's cool. Let's see. So we have flu and we have COVID, obviously. There's RSV. I think there's rotavirus. What else would we like? If there's no treatment, it's not worth knowing. So you could have a West Nile rapid test, but can't do anything. Yeah, you're West Nile positive. Because for Tony Fauci, they could have stopped giving him antibiotics, right? There's nothing else they could have given him. So the question is, should we have an antiviral for West Nile? Yeah, it could easily be made. It's just a matter of money. So you decide whether you do it or not. I think given those numbers and the fatalities and the long-term effects, I think West Nile vaccines and antivirals are warranted. And, there are companies that will pay for the development of vaccines. Nonprofits like CEPI, Coalition for Epidemic Preparedness and Innovation. They raise money for donations and then they fund development of vaccines that no pharma wants to get involved with, right? I think that's great. They're doing Nipah vaccines. They participated in COVID vaccines. I think that's the model going forward. We need to have that brought to development. And then once you have a vaccine, they also pay for phase 1 and 2. And once you're through phase 3, they can sell it to a company to manufacture. And it saves them a lot of development costs that may scare them otherwise. I think that's the way to go. So I'm not sure I want many more rapid tests of any kind. Let's see. We wanted to know about West Nile virus surveillance. Yeah, there's so state public health departments do surveillance, and there's a website called ArboNet. Okay, ArboNet. The clinicians send their disease there, and they accumulate it. But I know that there are websites where you could find West Nile virus surveillance in mosquitoes in different states. So arboviral surveillance, we look for human disease, and then that's where the ArboNet network comes in. And some states have West Nile virus testing of mosquitoes. New Jersey is one of them, I know. Oh, Steph is here. Welcome, Steph, another moderator. Thanks for joining us tonight. Appreciate it. Les wants to know if they need enough cases for a clinical trial to complete quickly. Well, you can set up your trials such that you have a vaccine arm and a control arm. You vaccinate them and you send them out into the world, and you wait however long it takes for them to accumulate enough cases for statistical significance, right? And so people, when they get a positive test, they'll tell you, and then you follow up. So you just have to have some confidence that you would get enough within the, a year or two. And looking at the West Nile numbers, I think any year would be fine with that. So it seems to me that this, and then if you want to involve other countries, so Tony Fauci said we have to involve other countries. Now, there's an issue with that, and that is if you tested a vaccine in Madagascar only, you probably wouldn't be approved in the U.S. You have to test it in the U.S. because people are different in different parts of the world. And so even combining numbers in different countries, I don't think that's going to cut it. But I do think you have to get the funding, and maybe CEPI can do that, and you can do it in the U.S. You have enough cases, or multiple countries. It doesn't matter. Farhad, I got COVID vaccine plus booster last year. Should we get the new booster? Please elaborate on the pros and cons. So the cons of getting a vaccine, a COVID vaccine any year, we don't know of any. Okay? But as Paul Offit says, taking a vaccine because you don't think it's harmful is not a good reason to take the vaccine. So I'll just pass that on to you. Yeah, I'm not aware of any negatives in getting a COVID vaccine every 6 months. But I prefer to see data that says it's clinically useful. And we usually don't have that data, or it's pretty sparse. I don't want to give medical advice, but scientifically, if you are under 65 and healthy, I think you would not need a booster because your T cells are going to kick in. By the way, this problem with long-lived memory B cells does not appear to be a problem with T cells, because although they didn't study it in that paper, others have looked at longevity of T memory cells, and they seem to be good for COVID. So if you're young, you will make a good memory T cell response and prevent severe disease, most likely, because you never know if you're healthy, right? You may have underlying issues that you don't know about. So that's a risk you have to consider. Or you could say, if you just had the 2 vaccines last year, if you test positive, you're going to take an antiviral, and that's going to give you over 80% reduction of hospitalization. So those are the options. It really depends on your age and health status. Some doctors seem to think you can only get it if you go overseas. You mean West Nile? That's too bad. That's not true. Obviously, right? Would you patent the sun? And Jonathan is quoting Jonas Salk, when asked, why didn't you patent the polio vaccine? And he said, would you patent the sun? which of course you can't because it's a natural resource, right? But yeah, no polio vaccine was ever patented. A polio vaccine is not a natural resource. It's something he developed and Albert Sabin developed, right? Doesn't occur in nature. But he's saying everyone should have it without any encumbrance. And I totally agree with that. But, health is a for-profit enterprise. Companies make money and they develop drugs, and they make money and they develop drugs. And they pay their CEOs way too much. $100 million salaries is ridiculous. $500,000 is ridiculous. Well, maybe half, but anything over $1 million is ridiculous. Sorry. You're just wasting money. But that's the way business works. But yeah, I don't. Of course, you couldn't patent biologicals, then no companies would invest in them, because as soon as they made something, another company would make it generically, and well, it wouldn't have to be generic because there would be no patents. So it's a conundrum, right? That's why nonprofits driving drug and vaccine development makes sense to me. Now, I admit I am very naive. I'm mainly a scientist. I have no business acumen or finance or law acumen whatsoever. But I feel that health for profit just ain't right. And that's why we get in trouble like West Nile. Yeah. Could GMO mosquitoes limit West Nile transmission? Well, in theory, yeah, right? Because you'd release sterile males and then eventually the population would crash. So then you'd have less mosquitoes to bite the birds. And, you don't have to have a zero mosquito population. You just have to reduce it to a certain point. I don't know what that point is. But in theory, that's what they've seen with dengue in Australia. It impacts the number of cases when you reduce the mosquito population by releasing sterile males. Yeah, mosquito dunks. You can use those. Those little things you throw in standing water or get rid of standing water, but you can't get rid of it all, I guess. And root. Well, you asked in Twiv why DeSantis trusts the weather scientists and not virologists. The answer is he doesn't trust weather science either. He deleted climate change references from all Florida state law. Yeah, I guess telling people to evacuate was the right thing to do, right? And so he used the science as an excuse. But you're probably right. Yeah. It's a good point. Oh, boy. What tissue temperatures does West Nile better replicate? What a good question. I'm guessing, 37. Yeah, it goes well at 37. No, you're talking about tissue temperature. Yeah. So 37 is a good one. Yeah. Yep. Jill. Unused car tires and littered plastic trash collect water, which is where mosquitoes lay their eggs. Even a tiny plastic cap littered on the ground can be a breeding spot. That's absolutely right. And many countries that don't have a lot of money, people still like to drink bottled sodas. And the caps, remember those old, well, they could be plastic or metal caps. They just throw them on the ground. They collect water, and they're breathing sites for mosquitoes. Yep. All these things. Yeah, for sure. Tom says a former lab mate of mine helped make those mosquitoes. He's a professor at UMiami now. Good job. It's always nice when people do well, right? There are tons of mosquito species, and they are pollinators, etc. Killing them all out, sterilizing to stop disease transmission seems risky. But what about gene drive to prevent biting part Well, in these release programs, they never eliminate all the population. It's usually a single species. So for dengue, you could target Aedes aegypti. That's it. And that's one of the questions. What's going to be the environmental impact of getting rid of even one species of mosquito? And so we don't know, right? Certainly, all of them would be a problem, as you say here, but I don't think that people are trying to do that. Yes, Dixon's book is called West Nile Story. Yep. Yep. So I guess all the spiders that look awful are really beneficial. Yeah, spiders are spiders are good. You shouldn't kill them. I mean, if I find a spider in the house, I try and carry it outside. I just think it's a good product of nature, and you don't want to erase it, right? So I feel. Bats, dragonfly, birds, frogs, and damselflies eat mosquitoes. Yeah, there you go. Building a better mosquito trap with Microsoft employees. That's it, TWIV 390. It's a very interesting episode. Cool tech. I'm sure it's improved by now. Should go check it out. Yep. Cheyenne wants to know if he says mask wearing maintains higher mucosal respiratory temperature. It could transiently because, yeah, when you wear those damn things, you get really hot. But I don't know. Has it been measured? Does mask wearing raise nasopharynx temp? Yeah, they do. Paper I see here, they increase temperature in the perioral region. Yep. Visto says Chinese government may not know about any deaths in the countryside from COVID. Maybe. Is mask wearing more in China? I don't know. Will, what say you? Did higher mucosal respiratory temperature protect against SARS-CoV-2 infection? Oh, because of the masks. I really doubt it. Hard to know, though. Nobody has studied that. That's a really hard study to do. Is there an international organization for vaccine development besides NIAID? Well, there's some disease-specific ones, right? But not really development. I mean, GPEI, Global Polio Eradication Initiative. It just focuses on, eradicating disease. You don't develop new vaccines. But CEPI, CEPI.org, is an international organization, a nonprofit that supports vaccine development. I don't know of others. There may be others, but that you can go to their website and check them out. Does osteoporosis impact the development of long B cells? Let's see. They actually, B cells are implicated in development of osteoporosis. But I'm not sure the other way around. But one seems to pre-order the other. You think long-lived B cells live in the marrow? They do. They're in the bone marrow. That's right. Thank you for your contribution, John. If you want to support our efforts to make people understand science a little better, you can go right here to the link. You can go to Venmo or microbe.tv/contribute, or below the screen, actually below the chat, I believe. Yeah, there's a little $sign in a box. You can contribute right here at YouTube. But we get more of your contributions if you go to Venmo or microbe.tv/contribute. And we thank you for your support. Thank you, John. John wants to know, how do the dendritic cells that pick up a pathogen know to come back to the lymph node? And how does the immune response know how to get back to the site of infection? So it's chemokine gradients that are attracting the dendritic cells, but they're trafficking regularly from one place to another. But there's always some kind of chemokine that's directing them. And then you get back to the site of infection. The site of infection is producing interferons and other cytokines, and that will attract the T and the B cells back there. Yep. Jill says, I wish we could prioritize clean indoor air, just like we once had to make clean tap water a priority. We wouldn't need to chase variants if we weren't breathing in everyone's gross air every place. Yeah, people take air for granted, right? You're breathing it 6 liters a minute. So, boy, if there's something in it, you're going to get it. Mm-hmm. That's a good question. Wonder why some people are more susceptible to being bitten by mosquitoes. Me being one of those people. I am never bitten. Never, never, never. I don't know why. It's some attractant, most likely. You are producing. Actually, we did a paper on this on TWIV a while ago. People who are bitten a lot make some volatile chemical that the mosquitoes like, and they identified it in this paper. So, what can you do about that? Maybe we could have you eat something that would change it, right? So that would be the goal of that kind of research. Tom says, did I misunderstand that it is a general property of human coronas that memory B cells are not established, account for reinfections? So we don't know about B cells, long-lived memory B cells. We do know that antibody levels drop very low and people get infected every couple of years, not only with common cold coronaviruses, but back in SARS, there was no protection. And well, we know that people from SARS, one, don't have memory B cells anymore, but they have memory T cells. And MERS coronavirus, the same thing. There's no protection, no long-term protection. Whether it's an issue with the bone marrow and long-lived antibody-producing cells, we don't know. People haven't looked for those common cold coronaviruses. Remember, at the beginning of the pandemic, a paper was published showing that there's antigenic variation among common cold coronas, and that was thought to explain the reinfection. But it may not, in fact, be just that. It may be the inability to establish long-lived antibody-producing cells in the bone marrow. Yeah. Yep. Yeah, so R naught is what you're supposed to calculate at the beginning of an epidemic where there's no immunity, no interventions. And then if you want to recalculate it, you have to call it something else. I forgot what it was, the new name. I'm not a fan of R naught or any derivative thereof, so I forget it. I don't pay attention. Does population density affect R naught? Yeah, sure. More people that are together. I mean, the people have to be together. You could have a highly densely populated state like New Jersey, and if the people don't get together, if they stay home, it's not going to be an issue. But look, people commute to New York, the most crowded city around, and on any given day, you encounter hundreds of people in a train, in a subway, restaurants, blah blah. So yeah, it certainly does affect R naught, or whatever the R naught is after the initial calculation. USA switched from Zostavac to Shingrix. Are the countries still using Zostavax? I think there certainly are. And I don't know about the UK schedule. Maybe someone from the UK can tell us. But there's a move in the US to switch the childhood chickenpox vaccination to Shingrix as well, because it's a protein-based vaccine. It's not infectious, whereas um, the chickenpox vaccine is an infectious vaccine and can cause some shingles later on, which is not good. What's your theory as to how West Nile came to New York? I think it was probably a rare person with high blood levels, which doesn't usually happen, and which we don't see epidemiologically. But that person flew over and in New York got bitten by a mosquito. It was August, lots of mosquitoes around. Thank you, Questella, for your contribution to science communication. We really appreciate it. And here's a promo for Environmental, a show Tuesday, Friday nights, 9 p.m. on climate and environmental. Check it out. John writes on Puscast, Daniel pointed out the Detict website, an app developed to 97% accuracy on identifying ticks and bites using your smartphone. It's very cool. Is there anything so conserved that a rat could test for microbes on Mars? Rapid antigen test. No, that's a good question. I'm pondering. I think there would be. Yeah. Yeah. Um, it depends where they evolved from, the ancestors. But if they're microbes, they're going to be similar, and have ribosomal RNA, they're probably going to be highly conserved. There'll probably be conserved parts of viruses. That's an interesting question, right? Let's get some samples and find out. Good for you. All right. Thank you, Environmental, for your contribution to science communication. We really appreciate it. G. Ferraro thinks the modified are better than spraying. Yeah, I agree. Heather is in Kamloops, BC. Welcome. Welcome. Is it a bad sign that Marburg is continuing to spread in Rwanda, despite that country having a significantly higher quality healthcare system than most other countries in Africa? Yes, it is, because even if it doesn't spread out of Rwanda, people are dying there, and it shouldn't be happening. We need to be. Well, there's no vaccine. That's the problem. There's some experimental vaccine that they're testing out, but this is a problem. Nobody developed a Marburg vaccine. Interestingly, the U.S. helped develop it because it was perceived as a military threat. It's the only reason it got to the point where it could be then tested during the 2015 outbreak, West Africa. So always an economic issue, right? But I'm not worried that this is going to become a global thing. I think their infection control isn't good enough. They need to work on that. Biological mosquito control is probably favorable to chemical. I believe the genetically modified mozzies only last for 2 generations. Well, it depends. The release of sterile males, they have Wolbachia in them that make them sterile, and then they can't mate. So once they're gone, yeah. There's no more. You have to keep releasing. Yeah. John says, I'm always excited to get a listener pick on Twib. If anyone missed it, there's a free education app called Cell Walk. iPhone only, but has a cell biology tour that you can spin around 3D. Since there's a West Nile vaccine for horses, wouldn't making one for humans follow a similar pathway? Well, yeah. The West Nile vaccine for horses, though, is a DNA-based vaccine. And these just don't work in people. So I think, you could make an mRNA vaccine. You could make a protein-based vaccine. It would not be hard. Nope. You've got to decide what you want to do. I'm guessing that trials for a veterinary project are not as expensive as those for a human. I think they're considerably less costly, yeah. But they're done. You want to protect your animals, right? Wouldn't it be helpful to have rapid tests for all known circulating viruses and bacteria so you can quickly interact with your doc and get appropriate antibiotic or antiviral? Yes, except there are not many antivirals, right? So if you get West Nile positive or rotavirus, there's nothing for you to do. I've always thought that we could have a test that would, you'd go in front of the mirror and the mirror would scan you and it would say, you're clear today, have a good day. Somehow a mirror technology developed that could scan you for all your infectious agents. Yeah, I agree. But in terms of being able to act on it, I'm not sure that it would help, at least not today. Maybe. So people always say we don't have antivirals because we don't have tests. We don't have tests because we don't have antivirals. So someone has to go first. Let's make the test. So, I do agree with that. Any idea if there's been studies on whether birds and or rodents get itchy after mosquito bites? I don't know how you do that, but it's funny. Do bats eat mosquitoes? I would think so. Yeah. Leave the bats alone. Yeah, bats eat tons of mosquitoes. Swallow and dragonfly bats all eat skeeters. And Ariosen said, Dixon's so clever. His title of his book, West Nile Story, right? You'd have to know the title of the Broadway musical. That's right. Okay. What professional activities could he have returned to if he had no mobility? Oh, this is 29293. Oh, yes, this was a patient that had severe infection. I don't know the answer to this. I don't know any background on that patient. Will says mask wearing in China has dramatically reduced. Healthcare workers are all required to wear them, but they're limited to surgical masks. During the huge surge in December 22, all N95. What's one thing you'd like to see from future research about innate immunity? I'd like to see if we can correct genetic defects in people that lack it, because they're underlying severe viral diseases, either not having the genes or making antibodies to the interferons. I think that would be great. I once stated overnight at a research statement, one of the female researchers had MS. All the mosquitoes went for her. Well, she may have had secreting some volatiles that did it. Yeah. Could West Nile mosquitoes take a plane? Sure. Why not? Siddhartha, thank you so much for your contribution to science communication. We really appreciate it. Can they make antibodies, antivirals in horses? Yeah, they used to do that. They used to make antibodies, right? In fact, I seem to remember that anti-tetanus toxoid was made in horses. But there are cleaner ways to do it now. You don't need to use horses. But Wayne says that we are doing it for sera for snake venom or some such. Yeah, maybe. Here we go. You know, you can make monoclonals now in cells. You don't need to use animals anymore. Okay, folks. Looks like we've reached the end of tonight. I want to thank our moderators for joining us. We had Les here, we had Tom, we had Steph, we had Barb Mac UK at the very beginning, we had Andrew. Thank you all for moderating tonight and keeping this a civilized discussion. Thanks everybody for coming. 134 people, 132 likes. Can't ask for more than that. Our next office hours will be a week from tonight, 23rd of October, 8 p.m. Eastern. Till then, folks, I want you to all be safe, take care of yourselves, and be nice. Thanks for coming. See you next time. Bye-bye.